Make sure we're recording here. I think I just started the recording a little late, but that's okay. So here's a poll real quick. There you go. All right, so let's answer that real quickly. There's some tricky wording in this one, but there's some things you can eliminate very quickly. So which statement describes the chromosome in a daughter cell at the end of meiosis? So you're looking for some keywords here. Okay, a few answers coming in. We've got about 25 people in today. So again, about 30 or 40 seconds to answer all these, and then I'll get going. If you don't get an answer in, don't worry, you're not going to get penalized in any way. Remember, it's just to try to make you better. Right? We're literally all over the place with this one to start. So I'll give you about 5 more seconds. Hopefully everybody can see that chat, and it just makes it easier for me to, if you throw those answer in. Okay, I'm gonna end it in about 3 seconds. Okay, so if you notice, I won't share all these with you, but you notice we're all over the board here. The correct answer, let's look at this. In prophase, I've got 24 chromosomes. So in prophase 1, what has happened up here above this? You think of, we go into something called, what are the steps before this? Give me to throw that in the chat and give me the answer to that one real quick. What would come before prophase, metaphase, anaphase, telophase 1? Let me throw that one in the chat real quickly. Hopefully these are things that you've just taught or just been taught. These are probably units that we've just done. So anybody? Yeah, that's right. Interphase would be the correct answer. So interphase would include synthesis. So what I'm thinking here, if have 24 cell or 24 chromosomes in that cell, it has doubled. So my original number was 12. So the goal would be to get that 12, multiply it to 24, because we're going through synthesis. At the end of telophase 1, that divides to 12. And then at the end of telophase 2, that will ultimately divide to 6. Okay. But that's not what the question asked. The question says, at the end of meiosis 1, how many do I have? So the answer has to be 12. So that automatically eliminates 24. All right. What else could I look to eliminate? All right. So this one says 12, this one says 12, this one says 12. What could I do to eliminate the rest of them? Each chromosome, what am I looking for, you think? So we're not looking at homologous chromosomes. That would be easy to eliminate there. Homologous chromosomes would only be in prophase 1. So that one's out. So we're on A and C, all right? Each chromosome is a pair of homologs. Each chromosome is one pair of homologs from the parent cell. The other one's found in the other daughter. So that's telling me that they have split apart because I get 2 chromatids in each one, all right? So ultimately the answer is C. Did I start you with a tough one? I probably did. But ultimately, you have to understand that we're reducing the number from 12 to 24 back to 12 to 6. Okay. So maybe we'll get a little bit easier as we go along. So the next part of this asks you to compare mitosis and meiosis. So mitosis ends with an exact copy. So we go from 2N and we end with 2N. And meiosis, we start at 2N and we end with 4 cells that have N. So some of those are going to look like my parent. Some of those are going to look different than my parent. And there's some reasons that we'll talk about a little bit here in a little bit about why those are changed or why those look a little bit differently. So some things about mitosis. Mitosis is 2N to 2N. It's one division. So this just happens once. It gives you identical daughter cells. So let's look at meiosis. So here's one round of division, and then here's another round of division. So there's actually 2 rounds of division. There's some things that cause variation up here, like I talked to you about and said, and I'll tell you what that is. That's crossing over that happens right there in the first phase. So it gives me some variation where mitosis gives me an original parent. All right. So you've got to know the difference between mitosis and meiosis. And if you can't say anything other than mitosis gives me an identical and meiosis gives me a reduced number of chromosomes to make a gamete, that would be okay. Okay, so let's see if we can pass that on. There's some quick things we can eliminate here with this one. All right, I'll launch the poll about 30 seconds. There's one disqualifier in just about every one of these that you should see except the right answer. Looking a lot better on this one. Okay, about 15 more seconds. Okay, very good on this one, very good. So the right answer is D, and the majority of us got that. Let's look at what disqualified in A. Mitosis does not make haploid cells. Remember, it goes diploid to diploid, so A's out. In mitosis, we don't have homologs. That one's out. In mitosis in C, we say, or this one says, meiosis doubles the number of chromosomes. That's not correct. We want it to be half the number so we can go through reproduction and get back to the full amount. So very good on that one. So that's just a little about meiosis, and we'll finish here. Remember, it goes through 2 divisions. So there's 1 complete division and there's 2 complete divisions. Meiosis, these are the things that I would hit. Meiosis involves homologs, and I'll show you a better picture of these homologs in just a second, but that's where the copied chromosomes from each original parent of this person will pair up and form tetrads. So tetra means 4, so that's 4 individual chromosomes that come together. So in metaphase, those tetrads or homologs line up. In anaphase 1, they pull apart. So you know meiosis 1 deals with homologs. It gets me 2 cells, okay? Meiosis 2 deals with sisters. So sisters line up, sisters pull apart, and that gets us those 4 cells. So the thing that happens, a couple of things that happen individually, you get crossing over in prophase 1. That only happens in prophase 1. It does not happen in prophase 2. Again, I'll tell you a little bit more about crossing over in just a second to get a lot more detail. Okay, that's going to increase the diversity. Ultimately, crossing over will, and then over here we get those 4 cells that are just a random combination of the parents. So that allows, in the end, that allows us to get things like random assortment into these different gametes. So the randomness gives us a bunch of different options. All right, we could go through meiosis multiple times and never get the random formation of these chromosomes the same, right? And then when you fertilize, you're going to do the same thing. You're going to get random in one parent, random in the other. So 2 randoms come together. So multiple different combinations of fertilization. All right. So here's what I was talking about with meiosis 1. That's where we get, specifically in prophase, that's where we get crossing over. So these homologs are going to pair up over here, and then they're going to attach, right? The place where they attach right here, and you see the little attachment point right there. So the point where they attach is the chiasma, okay? I'm sorry, it's not the chiasma, it's the synapsis. So the synapsis is where they physically attach, and then the chiasma is where they actually exchange information. So that's where they cross over. So let's look at this picture over here. If I look at these individual ones down here at the bottom, this one was originally like my parent. So it is exactly like the parent. It has no switching of genetic information, which would be called crossing over. So crossing over ultimately combines genetic information from this person's parents. So let's say blue is the original dad, red is the original mom. It's switching that information. So I get a little bit of both on those chromosomes. So this is a parent, this is a parent. These are called recombinants. So those recombinants are new genetic variations. They are not like either one of the parents. So this increases my genetic diversity as I go through the process of fertilization. All right. So um, this egg could be fertilized with sperm that would be recombinants. That is a different combination than the original parents, giving us new variation. So a couple of ways that we go through genetic variation increase. We get crossing over, they pull apart, so they segregate, we'll look at in a second, and then they make recombinant chromosomes. And then ultimately the combination of what's in this sperm and what's in this egg allow for fertilization, which would be a whole new combination of anything that the parents were able to give us. So increase in genetic variation. Okay, so let's look at this question here, talking about, you see meiosis happen up here, and then they line up and pull apart again. So that's two phases to ultimately create these four daughter cells. So I know this is really small. Hopefully we can see that and see what we get right here with the question. You can move the poll if you need to be able to see the question. Okay, so about 10 seconds. Okay, so majority of us picked the right answer, which was C. So these are going to pull apart, all right? So they pull apart in the next phase after is shown here, which would be anaphase. They do not come together. So nothing necessarily comes together. We don't have two traits that will always come together, which is allowing everything to pull apart. Separate traits go separately. So that's allowing us to see if something called independent assortment actually is going to increase that genetic diversity. So I said independent assortment creates genetic diversity. I said fertilization creates it. Crossing over creates it. So those are three big things that create genetic diversity. Okay, so let's switch to genetics a little bit. That's the end of meiosis. So all these are traits that, or all these are definitions that you should be very familiar with. I just threw them on there to say, hey, here they are. Hopefully you're very comfortable with those. Obviously, genes are found on chromosomes. Genes are going to help determine those traits. The way we represent the different gene variations are what we call alleles. A lot of times in genetics, we represent those with different letters. With dominant alleles are big letters, recessives are little letters. And then depending on the type of inheritance, which one of those will show up. The genotype is the genetic makeup. Phenotype is what you physically see based on the letters that you receive. You can be different combinations. You could be homozygous. If you get the two big letters, that's dominant. If you get the two little letters, homozygous recessive. So that's two of the same alleles as homozygous, two different alleles as heterozygous. Those should be terms that you've seen, I don't know, all the way since middle school. So those would be things that you're very comfortable with. All right. All right, so let's make this work. So these are monohybrid Punnett squares. So this shows that an organism is diploid. Okay, this purple flower has to have gotten a big R from its mom and a big R from its dad, and to make it purple. So it's only able, if this is diploid, it's only able to give either the big R or this big R. In this case, it's definitely given a big R. But what I'm trying to show you is we have a diploid organism and they can only give one of those. So in Punnett squares, we are showing the different options. showing the different options here. So obviously only given a little r, only given a big R, all 100% of these are going to be big R, little r, 1-to-0 ratio, purple to nothing else, purple to 0 white. Okay, all of them will be heterozygous. So we give a 1 here, we give 1 here to get back to 2, which is the diploid number. So that's the way you've got to shift and think about monohybrid Punnett squares. So mono means one trait, and that's, so we're crossing the two parents for the color trait here. All right, so we say this is maybe the parents, this is what we got in the F1 generation, and then you come over here in the F2 generation and cross those two hybrids or heterozygous. Well, if this is dihybrid number 2, they each can give 1. So this would be the purple homozygous dominant, this would be the heterozygous, and this would be the homozygous recessive. So we get a 3-to-1. This is one of Mendel's big ratios. Mendel, obviously you've studied Mendel, you know Mendel studied with pea plants, and he came up with the theory of genetics as we know it and study the basis of it. So he came up with a 3-to-1 phenotype ratio. So 3 purple, when I cross 2 heterozygous, you get 3 purple, 1 recessive white. Okay, and this is just diagrammed over here a little bit differently. But it's showing you the exact same thing to get that 3-to-1 purple ratio. So you should know the P generation is the parental generation where I cross 2 homozygous for traits. Then I cross the 2 hybrids in the F1 generation, and then crossing those 2 hybrids gives me the 3-to-1 genetic ratio. Okay. Again, if you have questions as we go along, let me know. And you can throw those into the chat, or anything that you want to focus on that I'm not, that's fine too. All right. So we know that we can represent these in different ways. So I can say there is a 75% chance getting purple. I can say that there is a 3/4 chance getting purple, or I can say it's a 3-to-1 ratio. Ratios seem to be the trickiest for you guys for some reason. Remember, ratios, the total has to add to how many there are. So that's not a 4-to-1, or it's not like a 3-to-4 like you would think of as a fraction. It's 3, it's just count them, how many there are. 3 purple to 1 white gives me the total number there. Okay, so we'll practice ratios again here in just a second. All right, so here's the 2 laws that Mendel came up with, and I've hinted at them a little bit. So law of independent assortment, we've actually already had a question about. So his independent assortment says that genes on different chromosomes do not travel together. So genes, 2 or more genes, like we got a big A and a little a, so dominant A, recessive A, got a dominant B, recessive B, they're going to pull apart in meiosis, specifically in anaphase, but they're going to pull apart, and they're going to move and travel independently of each other. So that means they never come together. Because I get the big A doesn't mean I get the big B. Because I get the little a doesn't mean I get the little b. I can get a variation of all bigs and littles there, or all dominant and recessive. So independent assortment means one trait is not inherited with another. So automatically you do not get one because you get the other, because they travel independently of one another. And because of this basis, that's how we get a dihybrid Punnett square. So if a monohybrid's one trait, a dihybrid has to be 2 traits. Okay, so the same thing, parental generation, F1 is the first generation, F2 is the second generation. So let's see how they travel. And I've got 2 options here. Okay, so we've got a yellow round with a green wrinkled. So yellow looks like it's dominant to green, because when I cross a yellow and a green, I get yellow. Round looks like it is dominant to wrinkled. When I cross the round with the wrinkled, I get yellow, all of them in that F1 generation. So 2 options. I'm either going to do a test cross. So I back cross it with a completely homozygous recessive, and I just get, well, let's talk about that in a second. Let's talk about when I cross 2 of these. Let's cross 2 heterozygous together. This is what I get. Now watch. This is the full genotype of that parent. Heterozygous for the color trait, heterozygous for the shape trait. If the 2n is 4, that's the full complement is 4 alleles. Then in a haploid number, I can only give 2. So I'm given 1 trait for the color, I'm given 1 trait for the smoothness or shape. Same thing with this parent over here. All right, so basically I'm just looking, how many different combinations can I make? I get 1 Y, 1 R. So big R, big Y, little r. I mean, you can go through and make all those 4 combinations. And ultimately, that's how I get a 16 block Punnett square. All right. The Mendelian ratio for that is 9-3-3-1. So I come up with 9 dominant yellow smooth, 3 yellow wrinkled, 3 green round, and then 1 completely homozygous recessive. All right. So why do we have to know those? We know these ratios to be able to say if there's deviations from the ratios. It's kind of like why we learn anatomy and physiology. We learn it the right way. We learn how it's supposed to work. If anything's wrong, we can figure out why it's wrong and how to get that person back into good health. Okay. So here's your ratios. If I were to cross it with just this recessive, all right, this one would only be able to give the big Y little r, and then we would have 4 different combinations here. So ultimately, you would just get 4 different traits. So one of the other ratios that you have is a 1-to-1-to-1-to-1 ratio when I do a test cross. And I don't have a picture of that up there, so it may be hard for you to visualize. We'll see that down the road here in just a second. Okay. The other Mendelian ratio, or the only other Mendelian law, is law of segregation. So you think that might sound a little bit similar to independent assortment. Independent assortment is them actually going separately into the gametes. Segregation allows for that because they're going to pull apart. So they kind of directly relate to each other. So homologous chromosomes are going to pull apart in meiosis 1, and then sisters pull apart in anaphase. I'm sorry, homologous pull apart in meiosis 1, and sisters pull apart in meiosis 2. So but that pulling apart allows for the segregation. If we do not pull apart, we'll see later that is non-disjunction. That's going to give us 1 too many chromosomes, and it's going to give us a chromosomal disorder. So the separation allows for genetic variation here. Okay. So here's our first genetics question. So if we read this, dark is going to be big C, and short is going to be big B. So our dominant traits are dark and short. How do I know that? I cross a dark short with an albino long, and when I get the offspring, they're all dark, they're all short. So that means dark has to be dominant to albino and short has to be dominant to long. So you look at the letters up here, big C would have to be dark, and then big B would have to be short. So it's asking you, what is the dark short-haired genotype in cross 2? So what does this genotype right here have to be to get this ratio? All right, I'm going to show you 2 ways to do it, but I want you to give me a guess first. Okay, about 15 seconds, please. All right, here we go. So I'll share those results with you. It looks like everybody is on the right track there. So here's the way to do this. Okay, you can either make a 16 block Punnett square, and what would the letters be to get that 16 block Punnett square? Or you could think about, what would the letters be if I separated these out and did a dark albino monohybrid Punnett square and then a short long monohybrid Punnett square. All right, so let's think about that real quick. I've got a couple options. Let me see if I can draw these real quick. So if I'm drawing these, what would be obviously the long hair? Let's do dark. So to be dark, what would you have to be automatically? What letter do you have to be dark? And it's going to be hard to see the difference between big and little c, so I'll try to do my best. You automatically have to be a big C to be dark, right? You automatically have to be a big C to be dark, okay? So both parents are dark, but it's basically, the question is asking you, what is that second blank? All right, we almost know for certain what this other one is. I've got one parent with short hair, one parent with long hair. So remember, short is what? Short is big B. So I'm big B something. And the other parent is long hair, so they would have to be completely recessive. So I know the question just asked you about the dark one, but let's go ahead and answer the short and long hair parent too, okay? What is the ratio? I'm just looking here at dark and albino. If I add those together, that's 6:2, and that comes back down and gives me a 3:1 ratio, doesn't it? So I should know how I get 3:1 ratio based on two parents. All right, if I come over here, there are 3 shorts, 1 short, so that's 4 total shorts. And then there are 3 longs here and 1 long, so that would be a 4:4 ratio. Or I could simplify that into a 1:1. So over here, I need a 1, something that gives me a 1:1 ratio, okay? So you all answered, the majority of you answered A. So in order to be a 3:1 ratio, I know I have to give big C, little c as a parent. Both of those parents have to be heterozygous. So that gives this here, here, and here. That makes it dark. And then this one is the one and the end that makes it short. If this was big C, for instance, no chance. Everybody gets a big C. Everybody's automatically dark. Okay. So you should be able to, through process elimination, figure out that it had to be heterozygous. So let's come over here and look at this one. We've already got this parent. So if I want to get a 1:1 ratio, this one, if I'm big B, everybody gets what? Everybody clearly gets a big B. So there's no way I could get the recessive phenotype. So what has to happen? This one has to be little b. So these here are the dominant, these here are the recessive, okay? So you've got to understand ratios, and sometimes the easiest thing to do is to, instead of working your way out through a dihybrid Punnett square, it's easier to make 2 monohybrids. And I know the question didn't ask you about anything over here, but I just kind of added that, okay? So let's move along. All right, so another question. All right, so look at my expected ratio. You're like, oh boy, I see what you're throwing at me here. This is a chi-squared, right? So all these say the calculated value of the chi-squared. So this is going to take just a second, but ultimately, what have I given you? I've already given you that expected. So I should see that 9:3:1 ratio. In a chi-squared, I'm basically writing my null based on the expected. So my null should say that the expected genotypes are not deviated from this 9:3:3 Mendelian ratio. Okay, if I reject that null, then I'm saying, yes, something else is going on. There's some other type of inheritance besides what I expected crossing those 2 heterozygous. Okay, if I accept it, I say, yes, I'm crossing 2 heterozygous Mendels in play here. Okay, or I failed to reject it. Okay, so I'm gonna give you, I think I'll give you 2 minutes here. Let's see if we can go back and figure out clearly, in order to get this question right, you're gonna have to figure out, is the chi-squared value 1313 or is it 924? Okay, and then once you do that, you can eliminate the other one based on the critical value, which you have to come up with from the degrees of freedom. So I'm gonna give you about 2 minutes here. I'll throw on the poll and use the poll timer. All right, so I'm gonna give you 2 minutes, see if we can work this one out. Okay, so I think we're struggling with this a little bit, and it might be time wise. So I'll give you about 15 seconds, and then I'll come over and help you with it. Okay, so let's end this one. All right. We're a little bit all over the place on this one, so we may be struggling with chi-squared. So maybe it's good we threw this in. Okay, so what we have to do is calculate the deviation. So this deviation right here, so a deviation, and the first step is observed minus expected. Remember, you get your formula sheet on the exam. Hopefully you've done enough chi-squareds to know to set it up in a chart like this. So observed minus expected. So check my math. Is my math good on all those? It doesn't matter positive or negative because the next step is that we're going to square it. Is my math okay on all those? And then the next thing we do is deviation squared, but divided by expected. So we're going to take 196. We're going to do 196 and divide it by 90. We're going to do 169, divide it by 30. You get the idea here. We're going to do 16 divided by 30. And then we're going to do 9 divided by 10. Okay, your chi-squared value is going to be a sum of all those, fractions. So you should come out to the A answer here. So it should come out to 9.24. Okay, and the rounding might be a little off, so it might be, might get 9.1 or something like that. But the answer is far from 9.1, is way, way, way from 13.13. So now we have to figure out which of these we pick. Do we pick A or B? If you have questions about chi-squared, there's maybe another one down here a little bit later. So we'll work that one again, or you might be something you can ask your teachers to review. Degrees of freedom is just simply choices minus 1. Okay, so choices minus 1. If I have 4 choices, degrees of freedom is going to be 3. Based on the chart, we look at the 0.05 column to find out So basically we're saying that's 95% chance that we're going to do what we say we do, accept or fail to reject or reject. So that's going to bring us up to 7.82. So this is just a number that we get off the chart. We can go even a step further and say if my chi-squared value is 9.24, that's going to be bigger. That's going to be bigger than my critical value at 7.82. So what I'm going to do is I'm going to reject this null hypothesis. So these differences in the observed are much bigger than what the deviations are bigger than what I could tolerate. So I'm going to reject that hypothesis that says this is a 9331. Okay. Any questions on any of that? Yes, that's a good point. You know, you can clearly get the critical value uh, very quickly. So just look at the chart and say, hey, I know there's 3 degrees of freedom. The critical value is 7.82. So at least that gives you a 50-50 shot if you're running quick on time. This is going to take you, you know, 2, maybe 3 minutes. You get about a minute and a half a question. So, you know, you're going to have to make up time somewhere else. So good point there. You can definitely say, you know, I'm get a 50-50 shot. I get it down. It's better than a 25% shot that you got on just starting out. So good point. All right. So again, this is our crossing over situation here. You see in the picture, that may be the first thing you're looking at, that these are actually crossed over. So what we create, remember, are recombinant chromatids. These that do not cross over here are called parental chromosomes. So the ones that have crossed over increase genetic variation because they're combinations of the parents. So that's why we call them recombinants. So those are the new variants. All right. So let's look at some things here. What I've talked about all along is genes travel independently. That's the whole premise of independent assortment. So independent assortment says genes do not travel together. Well, at some point in the curriculum, you should have learned that not everything behaves like Mendel thought. You know, Mendel was operating in the 1850s. Science has changed. We've learned, you know, different things about genetics. And one of those is genes actually have to sometimes travel on the same chromosome. If we only have 23 pairs of chromosomes, we have many more genes in that. Sometimes they have to travel together. So linked genes would mean the genes just travel on the same chromosome and they're going to be inherited together unless we somehow switch these and cross them over and they become on different chromosomes. All right. So what we can use is this recombination frequency. If you think about recombination frequency, just says how much recombination occurs. So a percent that cross over, could be determined by just looking at how many of them cross over the total. So you're looking at the number of recombinants divided by the total. That gives me the percent of these chromosomes that are recombinants, right? The other percentage would remain parental. And we can look at that to determine how close they are on a chromosome because we operate under this premise, okay? The higher, and I don't have an arrow on here, but the more crossing over that happens, that means the farther they are apart. I know I'm right on top of that, but that's okay. The farther they are apart on the chromosome. Okay. Conversely, the less crossing over would be the opposite of that. So I'm not going to type that. You can deduct that. The less crossing over would mean the closer they are together. So what that's saying is these are very close together. So if they were to switch and become on different chromosomes, the crossing over would have to happen in that very, very minimal space. Otherwise, if I said, you know, one gene was up here and the other gene was down here, crossing over could happen anywhere through that space and they could switch chromosomes essentially. So what we have to operate on is this percent crossing over. So we say the more crossing over, the further apart they are because crossing over could literally happen anywhere in there to get them in different chromosomes of recombination, okay? If they're very close together, there's just a very small spot in which they could cross over. So clearing all that off of there, what we're able to get is a percentage of recombination, and then that takes it and lets us see the map distance. So 1% crossing over equals 1 map unit. Map units are arbitrary units. They're not necessarily like 1 centimeter. They're not a certain distance. It's just an arbitrary unit that you would have to create. So if you said ultimately this was gene A and gene B, and we come up and say that there is a 4% crossing over, 4% crossing over, we would say that gene A and B would be 4 map units apart. That would allow us to map. That would allow us to map the gene's loci. That's just basically saying their position on a chromosome, okay? So we would create something that looks like this. We would say, you know, here's A, here's B. There are 4 of these little ticks, 1, 2, 3, 4. So they're 1, 2, 3, 4 spaces apart. There's A, and then down here at the end, there's B. So as long as you can figure out which ones are recombinants, which would be these 2 at the end, and tell me how many of those recombinants we have over how many total offspring you have, you should be able to get me the percent crossing over, which translates directly to the map units, which translates directly to their locus to tell me how far apart they are on a chromosome. Okay. So that could be that could be a lot of places that's asked. That could be on the multiple choice. You could even have to draw it out, and hopefully we're taking it all paper and pencil, and that would be something much easier to draw out than have to upload that if we had to take it virtually. Okay, so let's practice some of that. Okay, so here's your question. We got about 10 minutes left. So let's practice this question and see what happens with linked genes. Okay, so if you notice L, M, N, O, P, Q, R, S, T, U, V, all those are on the same chromosome. So we say they're linked, and because they're linked, they travel together unless we get some kind of crossing over here. So clearly this is depicting crossing over. What question would that be able to answer for me? So quick answer on the poll, please. There's a couple you can answer real quick based on what we haven't talked about. And so that narrows it down to 2 choices probably. And if you look at the data, that helps eliminate the other one to make it more obvious. Ten seconds. Okay. So majority of us picked A. I appreciate that, following along, and we know C and D weren't correct. We haven't talked about methylation, even though methylation is a type of gene regulation. We haven't necessarily talked about that here, so it's probably not the right answer. But we have talked about distance and how that relates to the crossing over rate. So crossing over directly relates to the distance. If they have a 40% crossing over between R and B will happen 40% of the time. That means they're 40 map units apart, what that would mean. Okay, so moving along. All right, so we can have, this is like an autosomal chromosome. It's just a normal 1 through 22 pair of chromosomes, and those are the genes that are on those. So you can have autosomal linkage, but you can also have sex linkage. So that would be a gene that travels on the sex chromosome. On the left, I've got my 3 female options. On the right, I've got my 2 male options. All right, the reason there's difference in numbers, all right, females clearly have 2 X's, males just have 1. So I can have two dominants, so I can be heterozygous, I can be a hybrid or heterozygous here, or I can be homozygous dominant. The male can only be one or the other. So if this is a recessive disorder, the male can either not have it or have it here. So they either get the recessive bad X or they get the dominant good X, if you will. So the option is this would be a normal female, this would be a diseased female, but in the middle you have an option called a carrier. Males can't be carriers because they only have one X. So these two X's give females two things. They give them the ability to be carriers, and they also give them a less of a chance to actually possess diseases that are sex linked. So you're going to see a higher probability of males being sex linked, disease, uh, diseased in general. Okay, so a lot of these can be represented in pedigrees. So in pedigrees, males are squares, circles are females. In this case, you can either have four options, all right? And we've talked about all those just in a second. We've talked about sex-linked, so it can be sex-linked dominant or it can be sex-linked recessive. At the same time, it could be autosomal dominant or it could be autosomal recessive. The first clue on this one is that all of these are males that are affected. So the darkened in ones are the ones that are affected. Because they're darkened in and I have a high probability of males, I'm thinking, well, that's probably sex linked based on what I just talked about. It's also probably recessive because it doesn't show up in this generation. It doesn't even show up in this generation here, and I still have an affected offspring. So the fact that it can skip generations makes it recessive, and the fact that we have a higher proportion of males makes it sex linked. Now, can I go back and check it to make sure if I have time? Yes. A time saver would be to learn some of those rules, like I just said there. Skipping generations makes it recessive. Probability, the ratio of males to females makes it more likely that it is sex linked. Okay. So we'll see if we can, let's answer two more questions. I've got two questions right here in a row. Let's answer these and we're probably going to run out of time for anything else. Okay. So this is telling you what the chi-squared value is this time. You do not have to calculate it. Okay. Okay. Looks like we're all on the right track here. So here's the two parents. Calico cats can only be females because they need a black gene and an orange gene. So that would have given the black spots and the orange spots. Males can't have both colors. They can only be black or orange. So here's the options here. So this parent's going to give either the black or the orange. This parent's going to give the orange or the Y, making it a male. So that's why I would expect a 1-to-1 to 1-to-1 ratio, right? My chi-squared is 4.6. There's 3 degrees of freedom, which makes my critical value 7.82. This is clearly less than 7.82, which means I can do what with the null? I cannot reject the null, automatically making it either C or D, right? Which one is the better option? The null cannot be rejected because the chi-square value is less. So this is basically testing you to see, do I need the chi-squared value less or more than the critical value to accept it? And I need it less to accept it. So the correct answer is D. All right, next answer, or next question. All right. Again, we're looking at ratios. So based on what I told you about time saving, look at the ratios here and see what we can come up with. So I've got 40 red females, 39 male red eyes, and then you see some other ratios there. So based on the fact that those ratios are fairly even, as most of you are saying in the poll there, this would be autosomal. Okay, so if it's autosomal, more females do not have the sepia eyes than males do, in a ratio that matters. There's two more, but this doesn't really matter. So because those ratios and phenotypes ratios are very similar, it's probably an autosomal trait for eye color. Okay, everybody did a good job with that one. Okay, so there are a few things we didn't get to. I had a few more questions about the 9-3-3-1 ratios, a few more questions there. Codominance does not apply like Mendel did. You can have two dominants like AB blood type. Incomplete is the blended phenotype when I get the two traits. So we didn't necessarily get time for that one. There's epistasis, which alters the ratio, which means one gene affects the other. There's polygenic, where multiple genes affect one trait. And I'm just running through that real quick to put that on the recording to make sure you could see that if you wanted to go back and look at it later. There's also phenotypic effects. You know, the phenotype could be affected by pH, or it could be affected by temperature. And then we have sometimes it doesn't work like it's supposed to. So when we have chromosomal problems like non-disjunction, we would get an abnormal chromosome number, and that leads to chromosomal defects like Down syndrome, which you have where you have an extra chromosome, like this person would here. A lot of times the ones that don't get the chromosomes are miscarriages, so they don't actually come to full term. Okay, so I appreciate you guys being with us today. I will hang out here just a second. If there's any questions, I'll pause the recording and give you about a 5-minute break here, so MaryBeth can take over and talk to you about cell communication. So thank you again. Good luck. If you need anything, please let us know. We'll be happy to help.
Focus a lot on the cell communication part. And then we will hopefully get a tie in a little bit of how cell communication affects mitosis, the cell cycle. All right, so with cell communication, is this okay, Robert?
Yes, it looks okay.
Okay, I'm sorry. For some reason, it's just like I can only see one thing or the other. I don't know what's going on there. Um, Anyway, so cell communication, there are three ways that cells can communicate. And the first way is through direct contact. So two cells would be directly touching one another to share that information. And I'm going to try to skip through some of these questions so we can get to some that I feel like are a little bit more important. But I feel like with, cell communication specifically, they're going to show you pictures. Excuse me. And I think if you can, and sometimes they're intimidating and you feel uncomfortable with maybe what they're asking, if you can just orient and say, okay, I'm looking at plant cells and think about plant cells. And that usually makes you feel more comfortable with what they're asking and helps you get to the correct answer. So we have two plant cells. Thanks to plasmodesmata, they have holes in their cell walls where they can actually share these chemical signals. So those blue kind of shaped molecules. So animal cells also can communicate through direct contact. And I just wanted to show you a picture of immune system cells. And so we have a host cell that has eaten a pathogen and it's presenting one of those proteins on its surface. And that's trying to communicate to our T cells. So this is one of our adaptive immune response cells that can then recognize and say, okay, this is what I need to be looking for and help take care of to get rid of this pathogen. But they are directly touching to talk to one another. So the second way that cells can communicate is through local regulators. So the cells will be close to one another and they'll send or release signals to nearby cells to communicate. And so the one thing that I like, sometimes they show you, so this is a picture of two neurons. You can kind of see the neurons here. And they have zoomed in. So if you see this little black square, they have zoomed in and we're seeing where they're trying to talk. So they're not directly touching. They are very close, but they're not touching. And so this is why it's a short distance. So what I would like to ask you, and for some reason, again, I'm having a hard time of seeing the polls and stuff. So we're just going to use chat. And so for my first question, what type of transport allows calcium to enter? So I'm going to show you. So here's calcium. What kind of transport is occurring to allow calcium to get into this neuron? So again, if you'll try the chat box, what type of transport is occurring to allow this calcium to get into the cell? Right, I did forget to point out, I do have A, B, C, and D. So again, I'm having trouble with the polls, so you can just write A, B, C, or D, or the correct term. All right, good job. So calcium is a charged ion, and so it's going to need a protein channel in the membrane to get in. And so with this cell communication, and if you didn't get into the details, that's okay about the nervous system, but sodium ions would be rushing in. Once kind of the sodium concentration builds up down here, that's going to be the signal for calcium to come in. And then once calcium comes in, that's the signal for these synaptic vesicles. So these are membrane-bound vesicles that contain a signaling molecule called a neurotransmitter. So when calcium comes in, that's going to be the signal for these to start migrating their way to the end of this cell. And so this will fuse. And so my second question, what kind of transport is occurring to release that neurotransmitter, to release those red dots? So again, you can use the A, B, C, or D in chat. So what kind of thing is occurring to release the neurotransmitter here? All right, so it looks like we're all in agreement. I'll try to move through so we can get to some writing and stuff. So exocytosis, we're trying to release a lot of that signal at one time. And then another common or question that I have seen come up in the past, you see this picture. And so the neurotransmitter to actually, so this cell is trying to talk to this cell, and it is going to talk by using this signal. And so once the red circle, the neurotransmitter is released, it's going to make it to this cell. So what type of transport, A, B, C, or D, is going to occur to get the red dot, the neurotransmitter, to this cell? So again, use chat. Which type of transport will get the red neurotransmitter from this cell down to this cell? So again, the neurotransmitter was released by exocytosis, but now it's got to make its way to this cell. So what type of transport is occurring to get this molecule from here to here? Okay, so we're split, it looks like, between D and A. And so when the neurotransmitter is released, that is exocytosis, so bulk transport of a molecule out, but then it's in high concentration here and it's moving to low concentration here. And so that would be diffusion. So no energy is needed. It's just going to move from high to low concentration. All right. And so the next example I want to give you of local regulation or cells talking through short distances is quorum sensing. And so this occurs in bacteria. I do have a link, and my Google slide will be available to you from that link that Robert showed you at the beginning of this, and I'll show you at the end again if you want to go through, and you'll have access. But Bonnie Basler, her research is kind of figuring out how bacteria talk to one another, and can we either stop them from talking or stop them from being able to hear one another so that we can fight bacterial infections that way, since a lot of bacteria are becoming resistant to the antibiotics we have. So it's about a 15-minute video. It is really interesting if you have the time to check that one out. So these bacteria, they are in close contact, and so they would be releasing the chemical signal, this green circle, and kind of just releasing it. And since there's only two, those signaling molecules kind of just diffuse away, and these two cells don't even know that they're near one another. But bacteria are going to start dividing. So they divide asexually. And so my next question for you is, what do we call the cell division that bacteria or prokaryotes use to divide? So if you guys will go to chat on this one. And tell me, what do we call the division that bacteria use to divide? All right, so at this time of year, I usually get my students want to tell me mitosis as well, but you have to remember bacteria are prokaryotes. Prokaryotes don't have a nucleus, which is pretty much what mitosis means. Mitosis, we're dividing the nucleus. And so prokaryotes divide by binary fission. And so once they do that, here on the right, we're seeing lots of bacterial cells. And so now all those cells are releasing the signaling molecule. And so the odds of them receiving that signal is pretty high. And so what they'll do, this signaling molecule essentially allows them to detect their population density. And once there is enough of them, then they can actually launch their pathogenicity on, you or I, whoever's infected with that bacterial infection. And so that's kind of how they can act together through kind of local regulation. The next slide I have is just, again, if you're interested, viruses also talk. So this whole past year, we've heard a lot about viruses. And so these are interesting kind of short articles on how viruses also talk to one another. And so the last way that cells can communicate with one another is long distance communication, and that's through the use of hormones. Hormones will travel through the blood to get to cells far away from where that signal was actually produced. So to get us really into what I feel like is the meat of cell communication, I would like you guys, I'm going to focus on this yellow square. And if you'll go to chat, can you tell me the 3 steps in a signal transduction pathway in the correct order? And then looking at this diagram, which number or numbers correspond to that particular step of the pathway? So we'll try to knock out 2 things with 1 question. So in chat, can you tell me the correct sequence or correct steps of signal transduction pathway, and then use the numbers or numbers that represent that particular step? All right, so reception is, I think most of us looks like we're in agreement here. So reception is your first step, and that would be number 1. So the cell is receiving the signal here. 2, 3, and 4 is going to be our second step, which is transduction. And then 5 will be our response here. So this is a very common example, I think, for hormones to be given with cell communication. And so insulin is a very important protein that tells your cells to take in glucose from the blood. So you guys did a good job there. We have already covered this kind of with the calcium. So glucose is a very large molecule, and so it also needs the help of a protein to actually get into the cell. The other thing, just looking at this picture, some things to kind of think about: if you are seeing multiple arrows, multiple steps are occurring here before we actually produce the protein they're going to insert in the membrane to actually get glucose into the cell. And then we're going to talk about some other components. We'll talk about this question over here in just a second. So with insulin, I'll tie in kind of the idea of feedback here. So most processes in cells, we want to have a balance. And so blood glucose levels is one of those things that we need to maintain a set level of. And so negative feedback is this process that can help us kind kind of helps a cell get back to that homeostatic set point. And that's, I think if you're seeing something mentioning negative feedback, that's what you should be thinking about. So it's about maintaining homeostasis or getting back to a set point. And so, say this morning before this session, you guys had donuts and orange juice. That definitely is going to cause your blood glucose levels to rise. And so when that happens, that's going to tell your pancreas to release that signaling molecule insulin. So insulin's going to go out. It's going to tell your body cells to take up that glucose and use it for cell respiration. It'll tell your liver to take it up and store it as a polymer glycogen, so that you can have some sugar later on if you need it. And so by doing that, your blood glucose levels go back to that homeostatic set point. And then, you know, later on the day, you get busy, maybe you forget to eat lunch and you're hungry, your blood sugar levels have fallen. And so that would be a stimulus to tell your pancreas to release a different hormone, glucagon. And then that can tell your liver, hey, you stored some excess glucose earlier. Can you break that back down now? We need it. And so glucose levels will go back up and you're back to that set point. So again, negative feedback is going to get you back to a set point. And I'll mention it again, a lot of times with negative feedback, depending on what processes you're looking at, it really allows the cell to save some resources and save energy too. All right. So I kind of want to just mention and recap biochemistry as well. So remember, if you're talking about any molecules, you want to think about their shape and their function. And so this very simple diagram for signal transduction pathway, we have a signaling molecule. It can also be called a ligand. And it is going to dock to a receptor that also has a shape for it. And so that's important to think about. So not all cells are going to have receptors for every signal that's out there. And that's important because they wouldn't be able to respond appropriately if they did. So when this signal docks, remember your receptor is a protein. And so I think that's probably stressed more throughout this class is protein shape is really important to the way that that protein functions. And so when it docks, this receptor, kind of just like an enzyme does with its substrate, is going to kind of hug around that ligand. So this receptor shape is going to change when these two interact. And that shape change on this kind of intracellular side of the receptor is what's going to initiate step 2, or transduction. And transduction is usually going to, this part can look a variety of ways, but you're usually going to see a lot of proteins. And in this picture, you're seeing that they all have different shapes. And that's what's going to help us get to a very specific response over here. So the one thing I would encourage you, if you're seeing a cell signaling pathway, and it, maybe looks different or looks unique, or it's very specific to a particular situation, the one thing I would encourage you to pay attention to is where's the response happening in that cell. And that's going to help you, I could ask you, make a prediction of what a possible response could be in this cell. And you could determine that by looking at its location. So if the response is happening in the cytoplasm, then we are either going to activate or inactivate a protein that already exists in the cytoplasm. And then if the response is happening, if you could imagine that a nucleus is here, then a very possible prediction of what the response could be is we're going to either turn on genes so that we produce a protein needed to respond to this signal, or we could stop those genes from being expressed so that we no longer make a protein. So pay attention to that. This is obviously a very generic picture of a signal transduction pathway, but those are common questions that I've seen come up. All right. So to get you guys back to biochemistry and some things, if you joined me back in January, we talked about this. So your signaling molecules can be water soluble or they can be lipid soluble. And that's going to kind of give you some clues of what could happen, but it also might help you answer the questions. So if they're telling you a molecule is water soluble, so let's go to chat. Do you remember two other names for water soluble? So let's start there. All right, so it looks like those of you that are answering are in agreement. So if it's water soluble, it is hydrophilic or polar. So those are other words they might use. And so why is a water soluble signaling molecule not able to cross the membrane? So that would be, if you'll put in chat, why can that water soluble hormone not just come into the cell and cross the membrane? So in this, you kind of want to think about properties of the membrane. Think about the characteristics of that phospholipid bilayer. Okay, anybody else want to try it out? So what I'd want you to be thinking about is think about the phospholipids. They have those hydrophobic tails that make up the interior of the membrane. So somebody's getting it as it's coming out of my mouth. And so they don't interact. So if this is polar, that's going to make it much harder to pass through that hydrophobic core of the membrane. And so water soluble hormones, you're seeing it dock to the receptor. I think that's important to pay attention to. It's going to help you kind of think about those ideas, you know, why it can't pass through the membrane. And the other thing I would want to stress is that signal is not coming into the cell. So it is going to dock to the receptor changes shape, and that helps kind of pass the message down until we get a specific response. So in the other box here, kind of just opposite lipid soluble hormone. So since it is a lipid or lipid soluble, your other names for it, I'm going to save us a little bit of time since you guys did so well on water soluble. That would be hydrophobic or non-polar. And so since the membrane has that hydrophobic core, it can very easily pass through the membrane. Its receptor will be inside the cell. So we call that intracellular receptor. And a lot of times, I feel like with most of these, that complex of the receptor with the hormone, the lipid soluble hormone, will act as a transcription factor and regulate gene expression. All right. So we're going to do a short answer and kind of think about the three parts. So with your free responses, that task verb explain comes up a lot. And so a couple years ago, we kind of had more of a generic picture, but I want you to use it with this one. So this picture over here with the growth factor. Reception, these were actually numbered. So it was like 1, 2, and 3. So I'm telling you what they are. And so the question wanted you to explain the role of each step in regulating gene expression. So if you're explaining, you're wanting to give me some details of how that step actually happens. So how would you explain reception? So we'll take it part by part. So if you'll go to chat, and how would you explain this reception step? What's going on here? Okay, I've got one answer. Anybody else want to try it out? How would you explain reception? So how would you explain this step right here? All right, so what I'm getting so far is good. So some of you are telling me a signal binds or attaches to a receptor protein in the membrane. You called it a ligand. Some of you, specifically use the word given. So a growth factor binds to a receptor. And that's great. That's exactly what's happening in reception. So let's move on. Transduction. How would you explain that part of a signal transduction pathway? What actually happens during transduction? Okay, so for transduction, I do have a few responses. Transduction can always look different. And so here in my picture, I do have a phosphorylation cascade. And so some of you are giving me specifics about that. It amplifies the signal. Um, the the thing that, and I don't always feel like I do the best job of telling my students this, is transduction is kind of that step that's going to connect the reception of the signal to the cell responding. So the signal is received at the membrane, and then transduction is going to help transfer, and that could be through amplification, transduces, transfers, however you want to word that, to the cell having a specific response. So to get the point here, they did want you to make that connection, that transduction is kind of that connection of the signal being received here to having a specific response. All right, so most of you did really good. That answered that question. And then the last one, so again, you want to focus on this picture where they're showing you response. So what's the role here in regulating gene expression? So if you are still thinking, I would encourage you, really look at this picture about the response. So what specifically is this cell doing? Some of you may not have made it to Unit 6 just yet. I'm in the process of finishing that unit up, so you may not be, all of you may not be here just yet. All right. So the key in this picture, we are seeing from this signal, this transcription factor is becoming activated, and then we do see mRNA being produced. So this signal said, transcribe this gene and make mRNA so we can eventually make a protein. All right, so that was 3 points. Reception, we have the ligand or the signal docking to the receptor. Transduction is going to help us take that message of the signal to having a specific response. And then the response in this picture is transcribing the gene so that we can make a protein. All right, so very good on that. I did want to just give you a picture. The pictures, I think all my pictures in the past have kind of given you a generic receptor. And so the membrane bound receptors can look 3 ways. So I kind of just like to show you these so you're not surprised or thrown off. So you can have a G protein receptor. You can have tyrosine kinases that look like this. And you can have the ligand-gated ion channels. So we saw this with the nervous system picture earlier. This was given with the insulin example earlier. And then this G protein is a great example with epinephrine, which I'm going to talk about more here in just a second. All right, so just to spend a little bit more time on the transduction part. So again, transduction can be a phosphorylation cascade. It doesn't have to be, but I do want you to kind of take a second and look at this picture. This has a lot of protein, so that's a lot of molecules and resources in that cell. And then you should be seeing ATP. So this means a lot of energy is going to have to be used to actually respond appropriately. And so kind of what I want to stress to you is we have our signal, binding to the receptor. What I want to point out is we have inactive protein kinases. And so to get through transduction, we're going to activate that. And so what I want to stress to you is that shape change. So it's kind of an oval-like shape over here. When it gets activated, it's now circular. And so kinases are enzymes that phosphorylate other molecules. So they add a phosphate group to them. And so something I feel like we are talking about adding a phosphate to another molecule. Anytime you add any other chemical molecule to another molecule, you are changing its shape. And so if you're changing its shape, you're changing its function. And so this continues, the phosphate is coming from ATP to activate all these kinases until we get to our cell response down here. So just to go back to negative feedback, sometimes if protein gets made here, once it's made, the cell has responded appropriately. And so that protein can sometimes come back and stop this from occurring again. So you might see cell signaling pathways showing you that, and you would need to recognize that as negative feedback. So what's that important? You're kind of shutting this down, all resources kind of get saved, all that energy not having to be used since we've already responded appropriately. In this case, we have protein phosphatases are another enzyme that would remove the phosphates and help us get back to the inactive form of those kinases. And so one other question that I feel like I've started seeing, what do you think the benefit is of having these inactive enzymes already kind of just hanging out inside of the atom rather than, going through transcription and translation when the signal is received? So what would be the benefit of having, proteins already produced and hanging out in the cytoplasm? So if you'll go to chat and tell me why you think that's a benefit. All right, good job. So a faster response is what that's going to allow us to do. So let me really fast, I'm going to talk about epinephrine. So epinephrine, again, just kind of showing you once the molecule is docking to a receptor through this phosphorylation cascade. Some of you use phosphorylation cascades can amplify signals. And so epinephrine, if you're not familiar with it, it can also be called adrenaline. So in times of stress or dangerous situations, that signal is going to help your cells produce a lot of glucose. So you can get 100 million glucose molecules released in response to that one epinephrine. So that's kind of what we're talking about when we say phosphorylation cascades can amplify a signal. So epinephrine is a great example of that. Epinephrine is also a great example of one signaling molecule. And then depending on what cell it docks to, what receptors available, we can get different responses. And so here, you're not seeing the whole process. We have the same receptor on liver cells and skeletal muscle blood vessels, but those transduction proteins are different, and that allows these two cells to respond differently. So if epinephrine docks to a liver cell, that's going to tell that cell to break that glycogen down and release glucose. Skeleton muscle needs a lot of blood, a lot of oxygen getting to it, a lot of that glucose so that they can fight or flee the situation. And then we finally have a different shaped receptor. It's not really shaped differently, but it is a different receptor on intestine blood vessels. And so if you're in a dangerous situation, you don't want your body thinking about digesting food. And so those blood vessels will constrict so that all the energy and all the oxygen is getting to cells that can actually help you get out of that situation safely. All right. Something I would like to do, this was a free response. I really like this free response. Sometimes I think when you're giving these cell communication questions, they look different than what you've had in class, and sometimes they can be very complex and intimidating. So what I would really always encourage you to do is kind of think about things that you have learned that is familiar to you. So that they are pointing out the cell membrane, so that should help orient you of what you're looking at. I'm seeing something inserted into the cell membrane. So, think about your cell membrane parts you studied earlier. So that could be a protein possibly. And then as I go through and kind of look, I'm seeing this caspase. I'm seeing a word end in ASE. So now when you guys reviewed with me with biochemistry, you knew that was an enzyme. So that's a protein. And that's going to just kind of help me think through this question. So I'm going to give you the information. We'll kind of walk through that, and then I'll show you the parts so that we can answer some of that with these last 15 minutes. So some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host. Host cells respond to these infections in a number of ways. So one of which involves activating particular enzymatic pathways. So we kind of already figured that out when we were kind of looking. And if not, they told you. So enzymes are proteins and they help speed up chemical reactions. So cells normally produce a steady supply of inactive caspase-1 protein in response to intracellular pathogens. So intra, that prefix means within the cell. So the pathogens already made it into the cell. The inactive caspase-1 is cleaved. So cleaved means cut and forms an active caspase. So here with step 1, we have the inactive caspase, and if the cell has a bacterial infection, we're going to cleave it and have active. So kind of pay attention to how this is different from here. Active caspase-1 can cleave 2 other proteins. So when you look at that, you're like, I have no idea what these molecules are, but they're telling you now that they're proteins. So that should kind of help guide you, what have you learned about proteins this year? When caspase-1 cleaves an inactive interleukin in step 2, so we also have a cleaving. So if you look at the difference here, the active portion of interleukin is released from the cell. And so just in my mind of what we've talked about today, I can see that they're showing me that arrow's going through the membrane. So it can pass right through that membrane. So it's either really small, hydrophobic, both. And if you don't know what interleukin is, they're telling you here. So an interleukin is a signaling molecule that can activate the immune response. When caspase-1 cleaves gasdermin, step 3, the N-terminal portions of several gasdermin proteins associate in the cell membrane to form large, non-specific pores. And so when I first looked at this picture, I was right. So I'm going to go to the question. So for part A, they want you to describe the effect of inhibiting. So we're stopping step 3 on the formation of pores and on the release of interleukin from the cell. So I want you guys to use chat. Describe the effect if we stop step 3 on the formation of pores and on the release of interleukin. So we'll go back to the picture. So what's going to be the effect if we stop step 3? So what's the effect on this pore? What's the effect on the release of interleukin? And so typically on free responses, you definitely want to use full, complete sentences, but here you can kind of just tell me what you think is going to happen in a shortened or faster version. So if step 3 is inhibited, what is the effect going to be on the formation of these pores and on the release of this interleukin? All right, so very good for those of you responding. So if step 3 is inhibited, the pore cannot form, and this had nothing to do with this step. So interleukin will still be able to be released or it will not be affected. And so that's 2 points that I think every single one of you earned. So I'm going to go back to the questions. So B is asking you to make a claim about how cleaving inactive caspase-1 result in activation of it. And then a student claims that pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection. Provide one reason to support that claim. So for charts, you're going to make a claim of how cleaving the inactive caspase activates it. And for the other part, a student claims that the pre-infection production of inactive precursors shortens the time for that cell to respond and provide a reason. So I'll go back so you can see. So you're making a claim of how cleaving this takes it from its inactive form to the active. And then you're given reasoning of how having these inactive forms shortens the cell's response. So I'll go back to the question again in case you need it. I'll see if anybody else responds. So B, we're making a claim about how cleaving the inactive caspase results in activation of it. So if we go back to this picture, if we look at the inactive, it kind of consists of two parts. You have this oval shaped circle and the blackened circle together. And to cleave it or cut it, what happens? White circle is still kind of the same, but the black circle is removed. So we've removed a portion of that. So we have changed its shape. And so changed shape activates it. Or if you said or were thinking that it removes the inhibitor, then you would have earned a point for that claim. The second part of this question was asking you to support a student. So the student in the pre-infection production of the inactive precursor shortens the response time. Can you provide a reason to support that? And so if you had said that cleaving is faster than making, so, the protein was already, all of these were already produced, all we had to do was kind of cut off that other component and it was ready to do its job. So that's much faster than making the whole protein. Or if you've gotten to unit 6, we're not having to wait for transcription or translation to occur to make that protein. So either one of those would have given you a point for that reasoning. All right, so I do want to let you have some data analysis, and it also helps tie in some of the other stuff you've learned this year. So researchers created a model using data from cell fractionation studies. And so they used specific proteins that they knew would be located in specific parts of the cell to help them determine information about a protein they didn't know. So this aconitase is a Krebs cycle protein. So you got to kind of think about, where is Krebs cycle? Where have you heard that before? So I'm going to try to save us a little bit of time. Krebs cycle, you should be thinking that's with cell respiration in the matrix of the mitochondria. DNA polymerase, again, if you haven't made it to unit 6, you may not have heard this, but if you're seeing DNA, where's DNA located in a cell? And so that's going to be in the nucleus. Glycolytic protein, so glycolysis, think about that. Where does that happen in a cell? So again, that's part of cell respiration. It's going to be in the cytosol. And then a sodium-potassium pump. Where have you heard of pumps being located? And so that would be in the membrane of the cell. So then we have this weird immune response cell that you probably haven't heard of, NF-kappa beta. And so I'm going to go back to our question. So a student claims that the NF-kappa beta protein is located in the cytoplasm until the protein is needed for transcription. So we want to justify that student's claim with evidence. And so the evidence was provided to us. So they're saying that NF-kappa beta is located in the cytoplasm until it's needed. So we want to go back. Where was cytoplasm? And that's here. And so this protein was found in fraction 3. And then they're showing us that this NF-kappa beta is also found in fraction 3. So that's our evidence. And that would give you 1 point. And so this part, I'll have you guys answer in chat. Identify 2 fractions where N-terminal gasdermin, so this is making us go back and look at that figure, where would that be found in cells infected with pathogenic bacteria? So we want to find 2 fractions where N-terminal gasdermin would be found in cells infected with a bacterium. So I'm going to go back and show you here. So here's our gasdermin. We want to find 2 fractions that gasdermin would be found in. So I'll give you a second to look at that. Where are you seeing it located in the cell? And then I'll give you this. And you can just tell me 1, 2, 3, or 4, but you want to tell me 2. So that gasdermin, which 2 fractions should it be found in based on this picture? So I'll show you the picture again. And there's your fractions. So you want to tell me 2. All right, so we are running out of time, and to finish up, this actually earned you 2 points for these, being able to identify the 2 fractions correctly. So that gasdermin, it started out in the cytoplasm, and we had said that the GAPDH was found in the cytoplasm. So fraction 3 is one you went with. And then if the cell has a bacterial infection, once gasdermin was cleaved, then it created pores in the membrane. So the sodium-potassium pump was also located in the membrane, and that's found in fraction 4. So 3 and 4, if you had said that, got you 2 points on that one. So that is about all we have time for. I do have the correct responses here to finish up if you want to use my Google slide later. Robert thankfully talked about mitosis. So I do have some information there and how communication can affect the cell cycle. You know, what happens when that goes wrong with like cancer? Why do they not listen to those signals? That was the rest of my information that I was going to share. If you have questions, you can always, feel free to email me on the introductory slides if you're going back. And then if you do want a recording of the video, let me on that slide. You can go here and then my Google slide will be attached. So you can get the rest of that or get those links about how bacteria and viruses talk if you're interested in that. You can stick around if you do have questions. You can use that to ask. But otherwise, I hope you guys have a great rest of your Saturday.